1 minute. Good afternoon and welcome to this 2023 Institute for Quantitative Biomedicine RCSB Protein Data Bank Spring Crash Course. My name is Stephen Burley. I'm the director of the RCSB Protein Data Bank and the founding director of the Institute for Quantitative Biomedicine, and I'm delighted to welcome you here today for this 2-hour session on the PDBx mmCIF Data Standard Data Dictionary. I'll begin by just providing a few introductory remarks concerning the Protein Data Bank, and then introduce my colleagues who will take you through the course objectives and then through the course itself. The Protein Data Bank was established in 1971 as the first open access digital data resource in all of biology. It started with just 7 protein structures. Some of the inspiration for establishing the PDB is shown on the right-hand side, with the first 2 protein structures, those of myoglobin and hemoglobin, and 2 of the first 3 enzyme structures, that of lysozyme, ribonuclease, and missing here is carboxypeptidase A. We've grown from 7 crystal structures of proteins to become the single global archive for 3D structure information for proteins and nucleic acids. And as of last night, we supported open access to more than 204,000 experimentally determined structures coming from X-ray crystallography, nuclear magnetic resonance spectroscopy, and increasingly 3D electron microscopy. The PDB is today managed by the Worldwide Protein Data Bank Collaboration, which includes data centers in the United States, Europe, Japan, and China, and then 2 additional members of the Worldwide Protein Data Bank Partnership, EMDB and BMRB, both specialist resources for 3D EM and NMR respectively. It's not an exaggeration to say that the Protein Data Bank has been walking the walk with respect to the FAIR principles since its inception in 1971. Recognition of this fact is found in our accreditation by the Core Trust Seal and more recently by the Global Biodata Coalition, that declared that the Protein Data Bank was a global core biodata resource. The RCSB Protein Data Bank is the US data center for the Worldwide Protein Data Bank. We're headquartered here at Rutgers University in Piscataway, New Jersey, with additional performance sites at University of California, San Diego, at the San Diego Supercomputer Center, and at the University of California, San Francisco. As a team, we convert global data into global knowledge. We are working with the outcome of the output of more than 60,000 structural biologists who have deposited data to the Protein Data Bank. 4 interlocking services take their information and transform it into knowledge through deposition and biocuration, which we do with our Worldwide Protein Data Bank colleagues. Management of the archive at RCSB Protein Data Bank here at Rutgers. The definition of the PDBx mmCIF data standards and integration of the PDB data with approximately 50 trusted external data resources. This information is in turn made available freely to 475 different external scientific data resources. We serve up this information on our data exploration web portal, which supports search, analysis, and 3D visualization of PDB structures. This is rcsb.org. And we also serve up much of the information that's stored in the Protein Data Bank archive in a training, outreach, and education web portal, pdb101.rcsb.org, that provides web-based in-person training, outreach, and education to millions of page viewers around the world. It's no exaggeration to say that the protein data bank impacts science ranging from agriculture to zoology. Reflecting that, our work is funded by the NSF, the NIH, and the Department of Energy. We support research in three key areas, those of fundamental biology, biomedicine, and bioenergy, as you can see on this graphic. rcsb.org, the homepage of which is shown here on the right, is a one-stop shop for access to public domain 3D biostructure data. In addition to the more than 200,000 experimentally determined 3D structures of the that come from the PDB archive that are delivered on the website, we also provide access to more than 1 million computed structure models coming from the AlphaFold database and model archive. These are the products of artificial intelligence and machine learning. Because the computed structure models use a data standard very similar to that of the PDBx mmCIF data standard, about which you'll be hearing a great deal today, the mmCIF data standard, the related mmCIF data standard, allows the PDB data to be interoperated with the computed structure models coming from AI and machine learning, so that we are able to use all of the rcsb.org functionality for searching, visualizing, and analyzing 3D structures of biological macromolecules across both the experimentally determined structures and the predicted structures, the computed structure models. So I urge you to use our website to get access to this information. Remember, when you are working with, when you do want to work with computed structure models, you have to opt in, you have to toggle on the include CSM button here on the top right of the homepage, and similarly on the advanced search page to be able to get access to both the experimental structures and the computed structure models. The advantage for you in terms of serving up the computed structure models is that it fills in gaps in the proteomes of the model organisms, many bacterial pathogens, plants, and also select microbial proteomes that are of great interest to the US Department of Energy. If you're interested in learning more about how we achieved this incorporation of the computed structure models with the experimental structures, please consult our latest nucleic acids research paper, volume 51, pages D488 to 508. So, having pitched the homepage of the web portal and the advantages of turning on the toggle to include CSMs, I want to now turn the podium over to Dr. Greg Critchlow, a member of the RCSB Protein Data Bank here at Rutgers University, and he's going to take you through the introduction and the course objectives, and then move you through into the first of the more detailed presentations on the PDBx mmCIF format and its utilization. Thank you very much for attending. Thank you for your support of the Protein Data Bank, and I look forward to talking to you again at the end of today's 2-hour session.
Thank you very much. Thank you very much for that introduction. As Dr. Burley just mentioned, my name is Greg Critchlow. I'm a biocurator at the RCSB Protein Data Bank. And I just want to briefly introduce what we're going to be learning in this course. After my brief introduction, we're going to have a presentation from Dr. Ezra Pazak, who will be discussing the conceptual framework behind the PDBx mmCIF format and the syntax involved. Following that will be Dr. Brian Hudson, who will be discussing how we can generate PDBx mmCIF files, how we can use them, how we can edit them in various practical applications. And following that will be a couple of presentations from Dr. Irina Persakova and Dr. Chenghua Shao, who will be discussing ways to extract data from file, analyze metadata from PDBx mmCIF files, how to do these manually via web portals, web interfaces, and also programmatically. And then Dr. Stephen Burley will be back at the end for closing remarks. So that's who we are. Now I want to discuss a little bit about who you are, who is in the audience. We noticed that we had a wide range of participants of varied backgrounds who registered. Most of you have deposited data in the PDB, but there's a large fraction who have not. And in terms of your experience with PDBx mmCIF, there's a wide range of backgrounds. Many, not quite half, but many have used PDBx mmCIF files for their research. Quite a number have heard about the format, but not very familiar with it. And some have never heard about the format. So people have a wide range of backgrounds. And in this course this afternoon, we have a little something for everyone. No matter what your background, you can get a little something for this course. I said this afternoon, it may not be afternoon where you are. So I want to begin with a little bit of the history behind PDBx mmCIF format. This actually was borrowed from the small molecule crystallography world. The CIF, crystallographic information framework format, is a format used in small molecule crystallography. And this needed to be adapted to be used for macromolecules to describe the kind of molecules we have in the protein data bank. And that was a major component of the thesis of the late John Westbrook, who described ways to expand the CIF format to accommodate large macromolecules. And he is actually shown here on the left in this top picture, and also way in the back in this bottom picture. But in the left of these pictures, that are pictures of the mmCIF working group at different times. So I do want to point out that this is not something that the Worldwide Protein Data Bank came up on with its own. Yes, the Worldwide Protein Data Bank was involved, but also many members of the community were involved. Here in the center in this picture, Paula Fitzgerald, who spearheaded the original working group project. And she formerly from Merck. And in these pictures, if you know some of the faces, you may know some people from industry, from academia, software developers, many different representatives from the macromolecular community who contributed to these working groups to develop this framework. So there was very close engagement with the macromolecular crystallography community to originally form these working groups. And what was developed was the PDBx mmCIF data format. And to the right, we have an example of a couple of sections I pieced together from a PDB entry, the PDBx mmCIF file from that entry that's taken from PDB entry 8C5M. And you notice it's obvious what this is concerning. At the bottom, you see coordinates X, Y, Z, coordinates, occupancy, B factor, the residue name. So it's a very readable file. The information we have for all the coordinates are here. This represents a coordinate file. You can have other files in PDBx mmCIF format. Also, the structure factor file can be in the same format. This is obviously a coordinate example. Since 2019, this has been the mandatory format for depositing coordinates from macromolecular crystallography structures. Refinement software packages can output coordinate files in this format. So it's very straightforward for the depositor to take what came out of their refinement software package and to directly deposit to the PDB. Mandatory deposition for cryo-electron microscopy and nuclear magnetic resonance, that is coming soon as the software gets ready for it. I want to note that today in this course, we'll be using the terms PDBx mmCIF, mmCIF, and CIF interchangeably. This will be strictly referring to the PDBx mmCIF format. That will be the proper definition, but we might abbreviate it for the purposes of this course. There are other flavors of mmCIF. For example, there's model-CIF, which is used to describe computed structure models that were alluded to a few moments ago. That's another flavor of mmCIF. So there are different flavors, but they all have the same core CIF structure in common. Since 2014, PDBx mmCIF has been the master archival format for the PDB. Has a lot of benefits. There are no column restrictions, so you don't have limitations on the number of characters you use, able to accommodate ribosomes and other large structures in a single file, therefore. You can realize the principles of FAIR, that is, that data in an archive must be findable, accessible, interoperable, and reusable. And the PDBx mmCIF data standard allows us to realize that. So an example on the previous slide, the data is very readable. You can find data you want. It can be parsed by computer, so you can access specific metadata that's needed, and you can reuse it, and it'll be interoperable in a variety of different platforms. As I just mentioned, you have different flavors of mmCIF. We use one flavor for experimental instructions of the PDB, another flavor is used model-CIF, used for computational structures, computed structures. So the data can be reused in many different platforms and many different websites, many different types of databases. And on top of that, it's fully extensible. So we can accommodate growth in the structural biology field. There's been a boom in structural biology in terms of the size of structures that are being performed, especially with cryo-electron microscopy, as well as new advances in techniques. The X-ray free electron laser has provided an increase in serial crystallography data and different CIF categories, and we'll learn about what a CIF category is in a short while, but different CIF categories were created to describe these. So we can see in this example on the right, which is taken from PDB entry 7Q7Q. And if any of the authors, we're taking these examples from PDB files semi-randomly. If any of the authors are in the audience, thank you for the work that you provided. We see in this example that this was delivered by injection. So we can read this straight from the file, and new techniques have been accommodated. Another thing about PDBx mmCIF is that it's founded on the DDL2 dictionary, and we'll learn about CIF dictionaries shortly, also in the very next lecture that will be presented. But what this allows is for checking of the PDBx mmCIF files for self-consistency. And then I mentioned it's human and machine readable. So these are the characteristics of PDBx mmCIF, and this is a very opportune time to learn about this for reason I'm about to mention. You see on the graph on the right, describes the 3-character chemical component IDs or ligand IDs, the 3-letter codes we use for our small molecules bound to protein structures or DNA ligands. These codes are running out, and these codes will be exhausted in approximately 1 year, possibly end of 2023 or beginning of 2024. Once that happens, any PDB entry that will be deposited after that point that contains a novel ligand or a ligand that is at least new to the protein data bank, those entries will be released exclusively in PDBx mmCIF format. So anyone who's interested in structure-based drug design, who's interested in ligand interactions, these new files will be exclusively in this format. So this is the perfect time to learn PDBx mmCIF. Also, currently, the PDB ID is limited to 4 characters currently. We'll soon need to expand that because we're running out of 4-character PDB IDs. When the 4-character PDB IDs run out, then all newly deposited PDB entries will only be available in PDBx mmCIF format. So that's something to keep in mind. So it's something important for users to learn and for software developers to learn. So just to summarize what I've said in this introduction, PDBx mmCIF is the archival data standard used for the PDB. The flexible and extensible format used allows for accommodation of new structural methods and very large structures. It can be read and interpreted by the human eye, but also can be produced and read by software. And we'll be learning more about these in the upcoming talks. Information from these files can be parsed programmatically, but also visually and by web links. And again, as I want to emphasize, shortly is going to, soon enough, this will be the exclusive distribution format for coordinate data from the protein data bank. So before I hand it over to the next speaker, I want to ask if there are any questions. And it looks like there are some questions in the chat. And I'm looking over to the chat now. If I see what's there. It looks like, well, some of the chat and some of the Q&A. Okay, so there are two different ways I see you can post your questions. So here we have a question from Guy Viggers. I often want to model missing residues in a structure by aligning an AlphaFold structure to a PDB structure, writing out a temporary file, and then using a text editor to simply copy and paste the missing residues into the PDB file, and then refining in Qt, for instance. However, since the mmCIF format is so much more flexible, how do I go about doing that now? Which sections of the mmCIF file do I need to edit, and how do I ensure that the same fields are present in the same order for both structures? Okay, so that is something that I think we want to come back and address later, because in the very next section, we're going to see the layout of the coordinate file in mmCIF format in more detail. And that will allow you to appreciate how the coordinate section, it's called atom site, the atom site section in an mmCIF file is organized. And knowing that will help to answer the question. So I'll defer that for a later point. And for time, there's there is another question from look through these, see what I have, because it's running short on time. Is there a template? Elke de Zitter, is there a template for the serial crystallography mmCIF entry, or did authors decide themselves which info to add? Okay, what is going to be added came from two sources, one from what's input in the deposition interface, and the other is recorded straight from the refinement package. Now, I'm not sure the depositors of the structure that I cited earlier. I'm not sure what refinement software they used and how much information was recorded there. Likely some of it came straight from the refinement package, but there are other ways, and we're going to learn soon about other modes such as PDB extract to capture data that are not captured from your refinement package, particularly if you're not using an mmCIF from your soft crystallographic refinement software. But I imagine much of that was captured from the crystallographic refinement software. And that will be the case generally for a lot of refinement methodologies. But some other things, though, were probably input in the deposition interface in our one-dep system. I'll read this last question. This looks like we're running short on time. Has a date been set for when the mmCIF file will be the only format for structures? Will legacy structures still retain the old format for download? The answer to the second question is yes. Previous structures will still have that the old format that is already available. No date has been set for when this will be the only format for structures. It's just a matter of time when we run out of PDB codes. We estimate we have roughly 5 to 6 years that we run out of PDB IDs. So that will be the time frame. It depends on how fast that happens, how many depositions we receive. And I said for the small molecule ligand codes, that will run out in approximately a year. So those specific entries will already be mmCIF only at that time. Okay, I'm informed that it's time to call the next speaker. So I'm going to introduce, but because some of these questions will be answered from the upcoming lectures, because some of them are very relevant to the lecture lessons that are about to be mentioned, that are discussed. And first will be Dr. Ezra Pazak, who is our PDBx mmCIF dictionary manager here at RCSB. And he's going to be presenting the format for us. And I'll turn it over.
Thank you, Greg. So I'm going to talk briefly in my time about the PDBx mmCIF data format and dictionary. I'll give you some examples of syntax and try to give you an understanding of the organization of the files. Hold on. So the first thing I wanted to point out, as been mentioned by Greg, is that the PDBx mmCIF data standard format is flexible. We can extend it as need be. It is human readable. The examples that you saw before, you could see that it really was a readable format. Even though it's designed to be read by machines, it is also available for humans. There are no limits in the width and precision of the data fields. So if we come up with a methodology which requires numbers that are wider than what would have normally been allowed in a PDB file format, we could accept that. The data values are controlled by a dictionary. The values must adhere to the various types that we define as regular expressions. Enumerations, limits that we are in the dictionary. I'll show you an example of that later. The relationships between categories are enforced by what we call parent-child relationships. And I'll give you an example of that, but that allows you to have multiple values linked up to one other category in the sense of, think of a citation. A citation, you have a single citation and you might have many authors. The data are organized in what we call categories, and they look like tables, and they're all the same set of keys and values. We can rapidly extend the dictionary and therefore the methodologies as scientific methodologies evolve. The dictionary is publicly available at this website, which I have just indicated over here. We provide tools to help validate data against this dictionary. And I'd like to just point out that this is the basis of other dictionaries. The mmCIF dictionary, which is used by AlphaDB and AlphaFold, use a format that's similar to the PDBx mmCIF with some extensions. So therefore, the core area is able to be read. The IHM-CIF dictionary is used by the PDB deposition system. It is for integrated hybrid methods. But again, the coordinate section is going to look very similar between methodologies. In all these dictionaries, we have similar information for instance, the structure, title, etc. So if you develop a methodology that could read one dictionary, extending it for some specific new data items in another one is relatively easy. So I'd like to, I'm sorry. Wait, what happened over here? Okay, so over here we have the website for the mmCIF dictionary resource. It is at the website mmcif.wwpdb.org. I'm highlighting in red information where you could browse the current dictionary, and at the top is a list of other dictionaries that are available. What's going on? One second, we're having some technical difficulties. How do I move to the next slide? Sorry about that. This page shows you the current, where our dictionary resource is available, and this is a website that describes it. So the management of our PDBx mmCIF dictionary, we are not the sole developers, we are not the sole creators of content and definitions. As Greg mentioned, it was developed in the late 90s by a group chartered by the IUCR CIF group to extend CIF for macromolecular data. We are one of the contributors as the WWPDB, but we also take input from the PDBx mmCIF working group, which is a group of experts in the field of crystallography. We also take input from developers who create extensions for their own dictionaries, plus we get feedback from users. Here's an example of a dictionary for pH of crystallization conditions. So inside this dictionary, we have what we call allowed ranges for the dictionary. For pH, you expect to be for most biological samples from 0 to 14. Those are not the, and you will not expect to see anything in our archive that violates that. However, we also provide for the depositors advisory boundary conditions. So some statistical analysis of all the structures we have deposited into the protein data bank showed that crystallization conditions tended to be have a pH between 3.5 and 10. Now, this doesn't say you cannot go beyond that. So there are examples in the archive of lysozyme crystals grown at pH 3.0. They're allowed, but they're unusual. We also have enumerations in the dictionary, and this shows an example of what we call the diffraction source, and we have a number of synchrotrons enumerated in here, along with their particular beam lines. This helps to ensure that there aren't typographical errors entered by depositors and allows us to have control of the data that's coming in and being put into the archive. So for the next few minutes, I'm going to talk about the syntax of PDBx mmCIF. As I mentioned, the data are presented in item value pairs, and we're going to describe an item is defined as made up of a category and an attribute values. So we have a category name and an attribute name. So sort of like citation would be the category, ID would be the attribute, and we'll just call this an item. Data can be represented in tables, as you'll see in the next few slides. And there are various rules for quoting text. So it is possible to have single quotes, double quotes within a text string. And we follow the same specifications as CIF. And if anyone's interested, here's a URL for it. But the important thing to remember is that our dictionary regulates the content of the public archive. So here's some examples. So we are showing for the structure of deoxyhemoglobin for HHB entry, here's what the unit cell is reported as. So it's a table of cell and various attribute names. So the length of the A-axis, B-axis, C-axis, the angles that are associated with it. We also have another syntax which is allowed, which is what we call loop syntax, which is you enumerate what the actual items are. So we have a name and an ordinal, and then you have a list of data values. This is a table, a tabular type of format. Both are equally valid. We could represent the cell length as a table in the same way, but it's not required. So here's 4 different examples showing the same type of text. And shows the different ways of showing either we have the upper left corner where we're talking about just items and values. We could have a more detailed quotation where it goes multiple lines. It's please life slow, snow. We could, as I said, we could loop it in terms of having multiple rows if need be in both syntaxes. So I would just briefly mention the parent-child relationships, as I indicated earlier, that you could have a citation, which we have an ID, which links back up to multiple authors. And we link these things together, and there are more extensive hierarchies available within the dictionary relating the various categories together. So within the experimental category, we have an entry ID. The cell we showed you the entry ID. We could have multiple chain linked together and in a form of a hierarchy. So to talk about the general purpose data file format, the thing that ties a lot of the categories together within the data files is the entity. The entity represents a specific macromolecule polymer chain. It could represent branched carbohydrates. It could represent individual ligands or non-polymers or water molecules. And you could see here that in this particular example, polymer entity 1 has a sequence linked together with the same entity ID. So in terms of reading the PDBx mmCIF sequence, this category, some people sometimes ask, what do you do for these non-single one-letter codes? We have an extended format where we put in parentheses what our ligand code that we're using. This is for a phosphoserine. But if you think about that, a phosphoserine is really just a serine molecule with another modification. We provide the canonical one-letter code, which would be represented by a serine. So if you're going to be doing a blast search, serine would be the one that you'd want to find. We have a mapping which shows how how the public author refers to residues. We will number things internally, and it's available in the public files in a sequential order. Bioinformaticians frequently want to know what is the sequence. I don't care what residue number the author chose. I just want to know it's the third residue in the sequence. So I show you this one. This is showing in the schema. We have the first one in the sequence is a histidine, but the author has chosen to use residue 311 in chain B. And this also depicts what is the proper alignment and registration for missing residues. So in this particular example, we are showing a large gap in the sequences for the structure. You could see that in 4LAD, there's a large gap in the structure represented by this dashed line, and you can see equivalently that the scheme shows there's a large region from 521 to 572. I'm missing the part in the middle. We're just missing, but we account for all the residues in the experiment. We enumerate all the non-polymers with its own separate scheme, where we could indicate what the author's residue number, which was 154, it was a terbium, and how we represent it. We also represent each polymer chain, each specific separate ligand with an asymmetric ID, which is used to give you an inventory of the different pieces. So we have a sulfate and acetate ions in the structure, as you can see from here. The coordinate section, as was already alluded to, is a little more extensive than the legacy PDB format. We retain moving from left to right, we have the full list of what everything is in column order, but I just want to highlight a few aspects. One is the ASIM ID. This is what is used. Every polymer chain is labeled from A, Z, and then double letters. Every ligand is labeled in a similar way to give you a unique one. So if you think about it, you could have within a structure 2 copies of the same polymeric chain. We'll call that, say, entity 1. We'll have the same sequence. They both have the same full sequence. But instance 1 might be, first instance might be ASIM ID A, and the other one might be B. We also, and as you could see, I mentioned the residue ordinal. This is the numbering from the start of the sequence, in this case 3. The author, however, chose to label it as chain A, residue 2. And the numbering of author residue numbers could be whatever that meets their scheme. We frequently have seen expression tags start with negative numbers. It's perfectly legal. The only thing we require is that there has to be an increasing value in going through the sequence. We have a way of showing intermolecular links using the strut-con category. And I just want to show how this, so this is a zinc finger, and I'm just showing that the metallo links between the zinc and the 2 histidines and 2 cysteines. We are seeing the residues 201, zinc 201, and how equivalently a metallo link. This is equivalent to the legacy PDB file format, and you're getting the same information just in a different place. The PDB format limits the precision that you could provide. So you could see a distance of 2.14 angstrom. And in the PDBx mmCIF file, we have 2.138. Since we do not have the limitations on space. So in summary, the PDBx mmCIF dictionary provides, and the tools that we have provide a well-regulated archive. And these two, the tools of the dictionary ensure that data are all conform to these formats. Our PDBx mmCIF dictionary is extensible, and we will make changes as experimental methods evolve and continue to do so. I covered the basic syntax, and I gave you some ideas of the high-level PDBx mmCIF model data. So I'll stop here and ask if there are any questions.
So we have a few questions in the chat. One of them is asking about, can you please provide a regular expression that represents the extended format for the extending PDB IDs?
Yes, I'll dig it up later, but it is available on the WWPDB news. I cannot give to you right on. I don't want to just type it in over here, but we can provide that information in the chat later, or later on send it to you.
So the link is in the chat.
Okay.
The next question is binary CIF only available? Oh, nope, Arena's going to answer that one later. So another question is, it's going to be alphanumeric. Thank you. What is the current, what about the currently first character?
For what?
For the extended PDB IDs.
That is undecided at this particular moment. The choice of the extended PDB, the important aspect, there are two things that were important. One was that we should be able to find within the literature anything that is a PDB ID. So that's why we have the PDB underscore prefix. And at the same time, ensure that we have extensibility into the future. I don't believe we have decided what the first character will be, whether it needs to be numeric. I think it is going to be numeric, but I'd have to double check that.
Okay, and we have time for one more question. Could you please elaborate upon protein ligand interaction?
Sure. So protein ligand interactions. Okay, so within covalent links to a protein to a ligand interaction, say a chloromethyl ketone inhibitor, you'll see a connectivity between in the struct-con category that physically says there's a covalent bond there. Other interactions that it might just be we're in the proximity of that is not recorded anymore. So if you're interested in knowing what is the environment around a water, our website does have some views of that, but we do not have recorded within this model file anymore. We used to have represented with site records, but we stopped using that about 4 years ago. The ligands in the rcsb.org website, there is a ligand section within the structure summary page that can give you that environmental view. But that's not accessible from within the model file. Okay, are we done? Or do we have more time for another question?
The questions are being answered in the chat.
Okay.
I think we should move on to
Okay.
Brian's talk.
So I will finish up here and say thank you very much for your time. Next up is Dr. Brian Hudson, who will talk about lifting the lid off the black box of the PDBx mmCIF files.
Thank you, Ezra. So my name is Brian Hudson, and I'm here to talk to you about basically some tools and tips to simplify the preparation of PDBx mmCIF files for deposition, visualization, and more. So how do you want to access, how do you want to use a PDBx mmCIF file? I think there are three general ways that people tend to use them. The first I call black box please, which is to treat the mmCIF file basically as a container. There's something in which you can move data from one application to another. You don't really care what it looks like inside, so long as it does the job that it's supposed to. The second level is about the same, but you also like to be able to edit some of the content. You do want to have some control over what's going on inside the file. And the third is I want to have as much control as possible, which is kind of what the rest of this course is all about, what Ezra's just been talking to you about, and what Irina and Chenghua are going to talk about here soon, which is for people who want to roll up their sleeves and get into the guts of the mmCIF file and move the furniture around, so to speak. What I'm going to be talking about here is mostly the first 2 points for people who don't necessarily or people who are in situations where you don't want to manipulate the contents of the mmCIF file manually, and you want to make some changes safely. So, first of all, I'm going to be talking in my talk mainly about PDBx mmCIF files in the context of atomic coordinate files. And the most important things that you tend to do with atomic coordinate files are produce them in one application or another and visualize them, because that's how we learn about what's going on in the coordinate files. And I'll go ahead and start here by just giving you what is essentially a partial list of major visualization programs that in fact read PDBx mmCIF file and have read these files for quite a long time. So if you've produced a PDBx mmCIF formatted atomic coordinate file, you will be able to find a place to visualize it. Secondly, the creation of PDBx mmCIF formatted files. The major X-ray refinement software packages all will produce mmCIF output. Phoenix and Buster by default, RefMac if you specify. There are various visualization programs that, in addition to taking PDBx mmCIF formatted files as input, will also output PDBx mmCIF files if you wish to. And then finally, there are the two programs that I'm going to be talking about primarily here in my talk, which are PDBextract and the PDBj-CIF editor. So what is PDBextract? Well, PDBextract is a data harvesting and integration tool that will parse data from multiple sources, specifically a coordinate file, data processing log files, and a template mmCIF file, and will merge all of these data into a more complete PDBx mmCIF output file that is suitable for deposition into the protein data bank. I've included the URL of the tool here on this page. Basically, what PDBextract does is it takes a number of categories from either a PDB formatted file or a PDBx mmCIF formatted file. For a PDB file, is a very particular categories that it will populate and it will actually ignore all the rest of the content of the PDB file. For a PDBx mmCIF formatted file, PDBextract will actually take everything that it can from it. And it will combine content from these multiple sources into a PDBx mmCIF file. And it will do so on a category by category basis. And this is an important point to make about PDBextract. Ezra introduced the sort of the nuts and bolts of the mmCIF format and how data are in categories. And PDBextract will populate these categories, but it will only populate a particular category from a single source. So it will not mix content from multiple sources into a single category. And it has a way that it prioritizes this content. And that is shown here. The highest priority content are the contents of the provided atomic coordinate file. as I said, either it can be in either PDB, the legacy PDB format, or in PDBx mmCIF format. You can also provide for X-ray, for diffraction entries, you can also provide reflection data files, and you can integrate, basically take all of those data files and integrate them into a single PDBx mmCIF formatted structure factor file. Actually, as a side note, this is a way that if you have multiple data sets but you can only provide a single structure factor file, you can take those multiple data sets and combine them together into one structure factor file. But the PDB extract will prioritize that information, and then it will add in additional information that is collected within the PDBx, the PDB extract interface, namely polymer sequences for various polymers within the coordinate file, and processing log files for diffraction entries from data collection and such. And then it will also take, and this is probably maybe the most important point I can make here, is it will also take the contents of a PDBx mmCIF formatted, what we call here the input template file. You do not have to provide this, but you can, and it's the way that you can get all sorts of other information into a PDBx mmCIF file. This can include things like contact author information, entry title, author list information, citation information, keywords, etc. Basically, anything that you can put into a PDBx mmCIF file can be included in this input template file and included in your output mmCIF file. And it will take all of these. It will any information that is present. Say, let's say you had you gave it a coordinate file, but you gave it also gave it an input template file that happened to have a set of coordinates in it. It would not use the coordinates from the template file, choosing instead to take the highest priority portion, which is the provided specified coordinate file. Let's see here. Oops, I'm going backwards. There we go. So this is the PDB extract page for the tool. Essentially, there's a lot of instructions here, and the place here where you can specify the experimental method that was used, the type of upload file, either PDB or PDBx mmCIF, and to provide the file itself. You press run, and you'll go to page 2. Up at the top of page 2, and this is also actually available on page 1, is the place where you can upload the input template file, the mmCIF formatted input template file of metadata that you would like to add to your PDBx mmCIF file. You can either provide it with a file, or there is also a link to the PDBJ, the PDB Japan CIF editor, which I will be talking about a little bit later here. Also in page 2, in particular for diffraction entry cases of diffraction, you can provide all this various information of various structure factor files that you will provide to integrate into a larger structure factor file, and various log files from processing programs. And then finally, at the bottom of page 2 of the PDB extract tool is the place where you can enter in the sample sequence. It will actually extract a sequence from the atomic coordinates that you provided, but here you can actually provide the full sequence should the sequence that was in the file have gaps in it. So by taking all of these here, I'll go back for a second here. But basically, this is just a way of taking all this information and compiling it into a single file, and to build a PDBx mmCIF file that has as much content in it as is possible. And because you can use this, you could, if you were making, say, multiple PDB depositions and you wanted to use a lot of the same metadata, author information, title information, et cetera, across a number of different depositions, you could just prepare a single template file for use with multiple sets of coordinates. Feed them all into PDB extract. So we also have here the PDBj CIF editor. This is PDB Japan. I've sort of inserted the PDBx portion of it here. It's a CIF editor. It's actually a general CIF editor. It's sort of CIF agnostic, as it were. It all depends upon the dictionary that you happen to be using. You can access the CIF editor 2 different ways, either via PDB extract on pages 1 or 2, and also you can access the standalone editor at this address here. You can use the CIF editor to build a new PDBx mmCIF formatted files. You can edit existing PDBx mmCIF formatted files, and you can check a file content against a dictionary to see if it conforms properly. It allows you to manipulate the PDBx mmCIF files safely, and you can either prepare content either for use with PDB extract, or you also have the option these days for EMDB depositions that do not include a set of coordinates, of providing a PDBx mmCIF file containing just metadata, so that information doesn't have to be input all manually in the deposition interface. So this is what the CIF editor looks like. There's a number of instructions on the page. The really important part is this little wagon wheel up here, icon up in the corner. That's where all of the action happens. Click on there, and it gives you the option to open an mmCIF file. Opening an mmCIF file, it will ask you for a dictionary. Now, if you're accessing it via PDB extract will automatically load in the PDBx mmCIF dictionary. If you're using it standalone, you'll have to choose a dictionary. It gives you several options to choose from, and you'll select a dictionary, and it will continue on. And it will show you the CIF content in the form of tables. And when it first starts up, actually, what it will do is it will actually check the contents of the CIF file against the dictionary you provided and let you know if there are any data values that are not conforming with the dictionary. So looking at these various tables, you can actually edit the contents of any of the cells in the tables. If you want to add a row to a table, you can just click on the name of the category, the name of the table, and you'll get the option to add a row. You want to delete a row, you simply click the X at the edge at the front of the row. If you go back up to the main menu and open it up, you'll get several different options available to you. One among these is an option called toggle tables. This is an option that brings up a list of the various categories that are available in the dictionary that you've loaded, along with a small description of them, allowing you to go and either deselect tables that are currently selected, or to select new tables to add to your PDBx mmCIF file. Going back to the main menu, you now have the option of either you can merge an additional mmCIF file, you can do the validation again. This is always recommended, validate early and often. And you can also save the mmCIF file for use either in PDB extract or in EMDB deposition, or whatever other application you might have for the CIF file you're working with. So before I go, I just want to talk, we've been talking a lot about PDB about deposition into the PDB. And I just kind of wanted to point out here, so to get a little deeper into the, open up the box a little bit more, what's the minimal, not to say minimal, but what information is absolutely required in a PDBx mmCIF formatted atomic coordinate file that you're attempting to upload to the PDB via OneDep. And I just list out here, and I'm not going to go through them in great detail for each of the major experimental methods, the various categories that are required. Now, it's important to note that this is what's absolutely required in a file, but it's not, you're not limited to that. And OneDep will require a lot of additional metadata for complete deposition. And under normal circumstances, if all you've provided is just the information that I've shown here on the screen, you will have to enter all of this information, all of the additional information, mandatory information, manually. And our advice, our very strong advice, is to make use of tools like PDB extract and the PDBj-CIF editor to prepare more complete PDBx mmCIF formatted atomic coordinate files for deposition. It will mean that you don't have to enter quite as much information manually, and you will be able to reuse the information across multiple depositions if you are in fact making multiple depositions. So that is all I have to say at the moment here, and I wanted to ask if there are any questions.
Okay, we have a question. You said that the PDBj-CIF editor can be used to prepare content for EMDB deposition. Can it be used to prepare content for PDB deposition also? Yes.
Insofar as you can, well, I mean, if you have a PDBx mmCIF file that you are considering depositing, just as is, and you want to make some edits to it, you can take it into the PDB into the CIF editor and edit it there prior to deposition. The other way to use the CIF editor is to use it to build a small, basically a small metadata mmCIF PDBx mmCIF file that you can then add to your PDB coordinates using PDB extract and build a sort of a bigger PDBx mmCIF file that way. Essentially, the PDBx mmCIF metadata file that you can deposit for EMDB is essentially sort of a more complete PDBx mmCIF file without any coordinates in it. Essentially, what it is.
Okay, we have another question. It's about privacy concerns. They're saying, concerning all the PDB related web tools, who's able to see the structures that are uploaded there for editing and validation? We're an academic lab working in collaboration with industry, and especially in the pre-deposition stage, privacy is a big concern.
To the best of my knowledge, all of these tools are entirely private. They do not retain any of the information that is entered into them, and yes, all confidential.
Okay, we have another question about mmCIF. There's nothing in the mmCIF documentation about cutting and pasting between structures. Do you know for a fact that it works?
Cutting and pasting between structures. Cutting and pasting between structures, it works if you're careful. You saw the mmCIF data. You saw the mmCIF structure, sort of the structure of the mmCIF format that Ezra showed, and Cheng-Hua will be showing you a lot more of this here when he gives his talk. Cutting and pasting between the structures, everything has to line up. So in other words, you have to all of the data items have to be as aligned with the correct data labels. If you've got that, it should work. But that's a big that's an if. So it's a very much on a case-by-case basis, I think, as to whether or not that will work.
Okay, I think it's time for our next speaker, Brian.
All right. So let's see here. So our next speaker is Dr. Irina Persakova, and she's going to talk about part one of programmatic file access and data parsing using PDBx mmCIF files.
Thank you, Brian. So in this part of the course, we will talk about programmatic file access and data parsing using mmCIF files. The next, please. So in the beginning, I will introduce what structured data files are available in mmCIF format, what's their content, and how to access the files. The next. So of course, the first in the list are atomic coordinate files that we already covered earlier in this course. The files contain a coordinate section with the XYZ value, residue names, numbers, atom names, atom types, chain IDs, ASIM IDs, temperature factors, occupancy values. The next, please. The atomic coordinate files also contain metadata information such as entry title, authorship, citation information, entry composition, source information, experimental details, data collection and refinement statistics, and so on. Also, the files contain derived and added information, for example, reference to the relevant UniProt sequence with annotation of sequence discrepancies if there are any, like shown in the example here. Information about the secondary structure, assignments, assembly information, unobserved residues, links, and so on. Next, please. The next type of files are chemical component files. The chemical component dictionary is an external reference file describing all residue and small molecule components in the PDB. The chemical component files contain the information about the ID, name, formula, weight, molecular weight of the component. They contain a coordinate section with two sets of coordinates. One is the model coordinates based on which the component was first created, and the other set is for a software generated ideal coordinates for the particular molecule. The files also contain atom connectivity section, as well as additional descriptors such as smiles and INCHI synonyms, if any, and additional identifiers. The next, please. Structure factor files, those are experimental data files for crystallography structures. They contain information about reflection indices, measured value of the structure factors and intensities, their sigma values, free flags, some metadata information. Note that those files might contain several data blocks. For example, one data block providing data used for the refinement. There might be additional data blocks used for phasing, unmerged reflection data, or anomalous diffraction data. Next, please. Validation reports are now also available in mmCIF format. Those files contain the same information as in the corresponding PDF or XML format files. They are well organized and easy to parse. they contain the information about structure quality at the entity chain and residue level. The next, please. Biological assembly files. Biological assembly is a macromolecular assembly that has either been shown to be or is believed to be the functional form of the molecule. The biological assembly files contain the full biological assembly for a particular structure, including all symmetry generated copies of each chain all within a single module with distinct chain IDs assigned to them as shown in this example. Note that the files might get very big, and check if your visualization software actually supports instantiation of assemblies directly from atomic coordinate files. This is just for better efficiency, for the efficiency. And next, please. Now, moving on to the data access, the main PDB archive is maintained by the www.pdb at files.www.pdb.org. The data are available through the HTTPS protocol. Note that the FTP protocol will be phased out on November 1st, 2024. The directory structure is, or rather a kind of a section of it is shown here on the right. Note that the atomic coordinate experimental data and validation reports are grouped by the two middle characters of the 4-character PDB identifier, like shown in this example here. And sort of similar logic applies to the chemical component dictionary files at the bottom example. The next, please. PDB also supports a so-called versioned archive. The versioned archive contains latest and prior versions of each PDB entry, or rather latest minor of each major version for each PDB entry. A major version number increment is triggered by the updates to atomic coordinates, polymer sequence, or chemical description, and minor version by other changes to the metadata. Note that in this case, the data are grouped using the extended PDB code, like shown in this example here. Also note that the PDB archived and version archive are updated on Wednesdays at midnight UTC time. The next, please. The rcsb.org also provides a variety of data access and download options. Shown here is an option to download multiple files at a time. The next, please. Of course, the files, individual files can be accessed through the download option at each structure summary page, as well as we provide a direct short style links for convenient download, as shown here on the right side. We also provide compressed binary and header only files, as well as chemical component dictionary files. The next, please. Of course, the direct URLs are convenient to use for programmatic file download, like shown here in using the URL lib Python module that we will describe more in details further in the course. The next, please. The rsync options are also available, which are helpful when maintaining full copies of the archive. The information is you can find it on www.pdb.org website. The next, please. As well as we provide the annual archival snapshots. You can access it using the link provided here on this slide. The example shows a section of the archival snapshot from 2018, for example. The next, please. We do provide the web services, the data API and search API services. For example, data API can be used to retrieve data when you know the PDB identifiers. The services follow the mmCIF dictionary, and the data are extended with annotations from external resources. We provide two interfaces, REST API and GraphQL interface. The data output is in JSON format. We provide tutorials that can help you to use the services. If you are interested to learn more about our web services, we are preparing a separate course dedicated to this topic. The next, please. So to summarize, the mmCIF format is flexible and extensible and can accommodate a diverse structural information available as atomic coordinate files, structure factor files, chemical component files, biological assembly files, and validation report files. And the PDB provides individual and batch file download options through the FTP, HTTPS, and rsync protocols. And that concludes my part of the presentation. Let me see if there are any questions.
So we have a question. Binary CIF only available at PDBE?
No, the RCSB I already mentioned also provide the option to download binary files. There was another question, can I download header only files? Yes, the answer is yes, you can download the header only files in mmCIF format. Will the RCSB continue providing an option to download PDB format atomic coordinate for all future depositions added using mmCIF format? So whenever the PDB format files, whenever we will be able to generate the PDB format files for the for the entries, we will provide an option to download the PDB formatted files. Can we please get the registration link for the web services related courses? Sure, as soon as we have them, we will advertise the links to those courses. Is there an identifier for a specific protein that can be used to programmatically pull down structures for comparison? I'm not sure if I quite understand this question. If you can specify, I'll try to answer that. Will mmTF files be maintained in the future or switch to BC files? I'll try to find the answer and answer this question through the chat. That's probably about it. So if you can advance the slide, please. So the next speaker is Dr. Chenhua Xiao, and he will talk about data parsing using mmCIF formatted files. Over to you, Chenhua.
Okay, thank you, Irina. Just in case if you answer questions, you can always contact us through our RCSB or email just to make sure you understand that. And if you have thought that our presentation was dry, and don't worry, I'll make it drier. So from now on, we'll talk about programming. So why? From Irina, you have heard that you can download either in batch or individually files from WPDB, RCSB, either FTP services. So, and you also learn from my colleagues, Edra, Brian, and Greg about how mmCIF file can host richer contents. And it's how it's defined in a way of strict item value mapping. And because of this rigorousness between the item and values, it can be parsed by program easily. And we call it parser. There are various mmCIF parser developed by the community and by us, and you can see some of them from our this page about the parsers in Python, in Java, in C++, in Perl, etc. But today I'm going to focus on the parser named just mmCIF, all lowercase, which is a primary parser used by us, by WPDB. It's a Python parser, but it wrapped C++ core. And it has the mmCIF dictionary API included so that you can verify the value against the dictionary. It's fast and reliable, and is freely available through PyPI and GitHub, as you can see here. So the reason we want to introduce this is, number one, from the pre-registration survey, about 40% of the attendants today have used the programmatic data access of PDB, but only about 10% had used parser, which is a very convenient tool to just make it work easier. And second of all, more than half of the attendees today have more or less used the Python, so Python parser will be welcome. You have learned some parsers from the Python scripting course about 2 weeks ago. That's from Bio PDB, and today's parser will be different. Today's parser focus on the speed and focus on parsing the metadata. So to understand how this parser works, let's review the mmCIF data hierarchy one more time. And at the top of this hierarchy, the data file which you downloaded from PDB, such as the one on the right, it's the data file for PDB structure 2H5V, which main contains multiple data blocks marked by the data underscore marker. In this case, it's data underscore and PDB ID 2H5V. And for PDB structure files, usually the contents are contained in the first data block. And one data block can have multiple data categories. For example, in this case, you see the top data category of entities, which means molecular components, and the bottom one, which is the data category for sequences. Data category is really the center of the mmCIF data file. It's the unit of data. And it focuses on the, as you heard from my colleagues, item and value mapping. In this case, as you can see from the sequence category, it has this data items of entity underscore polytype and add value of polypeptide for this protein annexin A2. And with the sequences in another data items of PDBx, sig underscore 1 underscore letter underscore code. And from now on, in the following slide, I will use a blue color to indicate its data item, and we use orange color to indicate its data value. So let's focus on the data category for the atomic coordinates category, which is item underscore site category. This is really the core of any PDB structure. And on the left side is what you see from the file. And if you kind of rotate the top of the data items 90 degrees, and you see a structure resemble the Excel spreadsheet, which you see like every day, not every day and frequently, and which also resemble data frame in some programming languages, where you see the top is a header or columns, whatever we call it, with data items, or attribute, which is the part behind the dot. Just remove the data category. And then you see the rows of values for each atomic coordinate. And from now on, I want you to just remember this resemblance and just the header and value, or the data item and value, and with this mapping. So just like you use any other Python packages, you use mmCIF in the same way. You import it and create a parser class instance. Then you read the mmCIF files. Then you can parse them into data objects. So there's 3 levels of data objects from parsed out of mmCIF parser. So first is the file level object, which is when you read a file, it creates a file level object, as you can see from on the top here. And it's called list of data containers, and which each container corresponds to the block level data object. And in the PDB coordinate file, it will be the first data block. And this data block will operate on the level 3 data object, which is category object. We literally call it data category. And data category is the key for this parsing and value extraction. Here are some just fundamentals of this, the usage of mmCIF parser. You first install, you pip install, you use GitHub to do mmCIF installation. Then under is IO, which means input output, and IO adaptive core is the one to handle the mmCIF files. And you create a parser instance, which is called IO, just for short. And then you can read a file, it automatically creates a list of the data container, which is a file level data object. Now, for PDB coordinate file, usually you took the first data block, which is the first of this list, is 0 index. And then from this block level data object, you can get all category level object. For example, you can use the method .get obj and given the category name, in this case entity, to get entity level data object. So if you don't remember, don't worry. All this code in the following slide will be in, they're already in this GitHub demo page, and I will show a short demo at the end of my presentation. So once you get this entity object, you can operate on it. On the right side, it shows this structure tree with entity category, which you can see that it has 4 entities or molecular entities identified by IDs 1, 2, 3, 4. And therefore, the 4 entities are, first is a polymer protein. This is protein is connectin A2. The second is a branch entity is for oligosaccharides. Then there's this non-poly entity, which is calcium ions. They're also the 4th entity of water molecules. Now, if you look at this entity objects data, you can see that it's basically a list of lists, which each row represents the row here. First, for example, 1 polymer, next thing 2. And if you want to focus on one particular data item or attribute, you can give the attribute name and use the method get attribute value list. Don't worry about the name, you see it in the demo, or you can download it from the demo. And you can get the 4 types of this structure, which is polymer, branched, non-polymer, water. And you can also do it, now you do it in the column way. Now you do it in a row way. So for the first row, next A2 entity, you can use the method get row attribute dictionary to get the first row's values as a dictionary, which the key as of the attribute and value of the values. Now, what if I just look at the cell, just one value, like a 19.2, I want to know the protein name. So you just give the attribute name and the number of the index rows. 0 in Python always means first, and you get this protein name in x22. So we have learned from my colleagues that the power, or you can say the complexity of mmCIF file lies on the interrelationship between the categories, because you cannot use one category for everything. In this case, in 70RT, author has indicated which ligand of this structure is their ligand of interest or subject to investigation. But the information about this ligand is in a different category. So how do we find, let's say, find for any structure, find what ligand are the subject of investigation or the ligand of interest. So what you can do is first you create the data object for this category for identifying of this ligand of interest using just like you get obj and give the category name. And from this category, you want to extract the component ID, which is ligand ID. And based on the ligand ID, you find information about the ligand in the other data category that defines this ligand. So in this case, you use a search function of select indices and giving the term you want to search, subject investigation, and giving the attribute of that for that term. And you get which row has this ligand of interest. And then you get a CCD ID, which is ligand ID of this ligand by using you can use this get value option function as well. And once you get this ID, you create another data category object for this lower data category. And then because you know the ID, you can use the search function again, giving the CCD ID you just identified and this component ID, this attribute, and found which row has, identified this ligand. And then you finally use the get value to get the name of that ligand by using the attribute name and that index. One more time on this data category is the core of the PDB structure is atom coordinates. In this case, you can just create a coordinates data object for the category for this mmCIF category. And if you want to know the structure, the model coordinates of that ligand for that ligand, you can select that ligand's name for the compound ID and choose then you get which row has this the coordinate for this ligand. Then you print out those rows based on each based on the attribute you want. In this case, you want, for example, the awesome compound ID, which is ligand ID. A sim ID is correspond to chain ID. Sequence ID correspond to sequence number, and this is atomic ID. Then X, Y, Z coordinates, as you can see the output here. Okay, Irina, I told you that you can also download the ligand definition file from PDB, and it's also in the mmCIF format file, and you can do the same with ligand definition. To indicate that some of the ligand have systematic descriptors, for example, SMILES, InCHI key, in addition to the general definition, including name, IDs, and type, formula, etc. So what if I want to get the InCHI key for this ligand KQF? So what do? You do the same. You import this adapter or mmCIF reader. Then you give the file and create this data container. Then you choose the first data container to the first one. And then you focus on this descriptor data category, getting this data object. And the other way of finding what you want is you should loop through each rows. This shows another way. You just loop through each row. This get row count means you are going to loop all the rows, and you create a dictionary for each row and try to find which one matches to InCHI key for their type attribute. Another type of mmCIF files is for the validation report that you can download it from PDB. And same thing in the right side is validation report fragment for PDB ID 7R2Y, which is the general geometry summary. And for example, if you want to get a percentage Raman-Chern outliers or the class scores, what you can do is you first read this file and create data containers, and then you choose this category as the data object. Then you find corresponding value for the corresponding data attribute or data items. And you get output here, as you can see. So I'll just give some fundamental introduction of how to use mmCIF parsers. And we all know recently like AlphaFold 2 kind of stimulated significant interest in the computed structure models, in short, CSMs. And mmCIF file standard provide data representation for the describing CSMs. It was developed by the mmCIF working group, and we are part of it. And this is standard used by the model archive and model-based repositories. Because it's an extension from the PDBx mmCIF dictionary, so you can parse these CSM models with mmCIF parser as well. And this just shows how to do this CSM searches on rcsv.org, which Stephen already gave. So you first turn on this include CSM marker. And then, for example, type NOVA on the top, it will show the molecules with this keyword. And for example, NOVA alternate splicing regulator 1. We do have fragment structures of this protein, but not full protein. And in this case, the structure prediction from AlphaFold DB provide a full prediction that we give access for you to download from there. And there's all these various colors for different confidence, which you can study by yourself on our page. And again, because it follows mmCIF data standard, you can use mmCIF parser to parse the CSM models. Just a quick summary of what I've said. You can learn about PDB mmCIF dictionary at this website, our main website, and there's the software-specific resources are listed under here. And mmCIF parser is the one we use primarily on parsing PDBx mmCIF files. It's good and reliable and fast. And you can see the demo here. Now, in the demo, what you will see is whatever you have seen from my slides, those quick introduction, you also can find 2 case studies. We have learned how to do cross-category search, but you can do cross-file search as well. For example, you want to know get a ligand coordinate from a coordinate file, and for this particular ligand, you want to find the chemical descriptors, and then you want to look at the validation report to see how good this ligand fit into the electron density and how good its geometry is. There's a case study number 2, which was a real request from a user that he wants to separate a set of PDB structure based on the species, which you can do. And now I've just switched here to a demo. But before that, I want to give you an example of real mmCIF files. We have all learned from our colleagues how to do this. I mean, but until now, I would say a real one because there's a significant percent of people haven't used mmCIF. And as you can see here, this is our main website, rcsv.org. As you can see here, if you want to search CSM, we can turn this on. Obviously, we want to turn it off for now for experimental data. So if you want to look at structure like 2HIV, I just talked about, you just type 2HIV and it goes here. Our short summary with various tools you can know. I mean, for example, ligand interaction. There's something about sugar ligand interaction you talk. One attendee was asking, but now we just want to look at the files. Now this is real PDBx mmCIF file. It started with data block, with the main data block data underscore, then PDB ID. And the first category is very short, very small category of entry. And with one item entry ID, which is PDB ID. Now, if you go down, you come to the entity category. Let me just make it bigger so you can understand. Because it's entity category. And just like I showed on the in the slide, there are 4 entities: protein, oligosaccharide, we call it branch entity. We recently remediated all the carbohydrate entities into branch because they can make branch, and the non-polymer calcium ions and water molecules. And if you want to focus on the sequence, these are the sequence. There's only one protein in this structure, so it's easy. There's only one entity ID, and this is the sequence for this protein, annexin E2. This is structure was resolved by myself when I was working in the lab. So now, this is the centerpiece of the PDB structures, which is model coordinates, mmCIF category. You can see that the XYZ coordinates, occupancy, B factor. It resembles PDB somehow in the layout, but now you have this strict atom value pairing. So now I'm going to show you, this is the demo page on GitHub, and there are a brief introduction about this. It's just very brief I put together. And there are cases about this very simple, simple, just like I just introduced in the slides. And there is this small download file utility you can use to download the PDB coordinate file, ligand file, validation file, which is easy to use. And other things. And the track LOI is the case study number 1, as I just said, and sorted by source is the case study number 2. You can just kind of browse by yourself and explore. But now I'm going to just do the first one. I made the first example, the simple Jupyter notebook. So in this case, as I said, you first import this thing. So you import, OS, like just for operational system. And the second import is for file download, as you probably know. And third import is one introduced multiple. This is the one from mmCIF for reading and parsing file, mmCIF files. To run it. Let me just make it bigger again. You can see better. Now, you start download the files. In this case, you give a URL. In this case, our URL is for RCSP, it's this simple URL you can download from PDB, which will be used from the download file, that small utility I just told you. And you give a name of local file name. If you want to download this, I just want to download this file to temp directory as the same file name. And then you just use this simple method URL retrieve, give the URL and local files, and we'll do. So it's the right downloaded file here. Now, you want to import this introduced IO parser instance, and you pass this file you just downloaded. So as I said, for PDB coordinate file, the data are mostly in the first data block. So you just choose the first data block, which is the first index number 0. And then you want to get the entity from that data block. Use the method get obj. So now you have the data object. Now you look at the data. So this is data. It's a list of lists with each element that this is solved to record the row of the mmCIF file, as I just showed you. Now, what if I want to look at the first row? So first row is, you have seen use this method get row attribute a dictionary, you get the dictionary-like things, just like any other Python object with the ID, value number 1, and type polymer and protein name annexin A2. And you can also just get the row of interest. If you know the index, you know the mmCIF item. So in this case, I just want to know which protein is it. And the first, which is first protein is annexin A2. And you can do the same for sequence, just like we run here. Create a data object for the entity poly category, and then just run it, you get a sequence. So again, if you have any questions, feel free to ask, and I'm going to switch back to the presentation. Yeah. So today we have learned how to understand the PDBx mmCIF mmCIF dictionary, how to read mmCIF formatted file, how to edit the file, and how to prepare the mmCIF file, especially for PDB interpretation, and how to access and parse data programmatically. I hope it has been helpful. Any questions?
So we have two questions. The first is, why is the order of the atoms in the alpha-fold structures NCACCB O rather than NCACO following the CCD order?
This is more, I think, convention they try to follow. But as I said, the order doesn't matter for mmCIF file. That's the key difference between PDB and mmCIF. There's no need to align the column, no need to figure out the ordering. You have this item and value mapping everywhere, and it's followed strictly and rigorously. So it doesn't matter, I think.
Okay. The next question is, what minimal categories have to be set for a .cif file, assuming in parsers like Gemi/BioPython?
If we want to talk about other programs, that would be more up to the developer for the software to decide. And for us, we have, if you look at mmCIF dictionary on the website, it describes what data are mandatory, what data are better to have, what data are optional. And it's almost, because there are hundreds of categories, it's almost impossible for me to give you, go by one, but you can get information there. And if you have questions, feel free to ask.
Okay, yeah. So his follow-up question was, he's asking, since he makes files, quote unquote, by hand, and a small amount of atoms for debugging, with a small amount of atoms for debugging.
Okay. Yeah, I mean, if you do that way, I guess you have to try by yourself. Especially if you use third-party program, I cannot answer just, I mean, in a general way. It depends.
Okay. Another question is, if I have questions about tags in the mmCIF, who should I ask?
Oh, you can contact us. Okay. So when you go to RCP, you can always contact us. There's a contact page, you can contact us. And you can also, I think mmCIF also have a contact information that you can use. Just reach out to us and we're more than happy to answer any questions.
Okay. Our next question is, can I use mmCIF files in R or PySpark?
Wow. Right, you reached the right person. I use R myself. I would say better don't use it. You better use process data for R, especially because R is just not very good in tracking back issues. There are probably programs or plugin developed by people, but R is really a data analysis program, and it's supposed you have some data pre-prepared for input. And for the other program, I'm not so sure. Sorry.
Okay. And then another question is, where can I check the minimum for mmCIF? They're asking, can they find that on the GitHub?
I'm sorry, minimum?
The minimum. Yep.
Minimum. Again, look at our mmCIF dictionary. The website I just look at here. I'll go up a little bit. Just, we try to get all the information. Sometimes the information is a little bit dry, and but if you want have any question, just feel free to reach out to us.
Okay, and our last question, we have a two-part question.
Okay.
You show a ligand definition file. Is that in the PDBx mmCIF coordinate file, or is this a separate file? And if it is separate, is it also in PDBx mmCIF with the same dictionary as the coordinate files?
So it's an interesting question, because for us, at this moment, the PDB model coordinate file do not have the full definition file included. And it's a separate file under a CCD archive. And as said, as Irina indicated, you can download all this, like more than 30,000 definitions from PDB and parse all of them and find a correspondence between this ligand file and PDB model coordinates. And I do know that some refinement programs such as Phoenix will write the ligand definition file at the end, sometimes resemble this was doing. And we may think about it, but so far they are separated.
Okay, thank you, Chunghwa. That's all of the questions.
Okay, great. So next, I will give back to Dr. Burley to conclude the press course. Thank you very much.
Thank you, Chunghwa, and my thanks to all of you from the United States and around the world who've remained online through the last 2 hours. So I'll just make a few closing remarks and then thank the many people who made this possible. So I'll begin by thanking you, the audience. We'd love to get your feedback so that we can make the next webinar in this RCSB Protein Data Bank series even better. So please take the Zoom exit survey, and you'll then receive by email a link to the crash course presentation slides in PDF format, and you can use this QR code on your phone to get to the easy to fill out survey. So please do that. We can't get better without constructive criticism. I want to advertise the fact that we have considerable amount of training information resources on our PDB 101 website. It's 101 for those of you outside the United States, means introductory course. So this is pdb101.rcsb.org, and if you go to the train button, you'll be able to get access to materials that will help you effectively use rcsb.org tools to search, visualize, and analyze 3D biostructure data. Among the things you'll find under the Train tab are training courses, the Guide to Understanding PDB Data, our Education Corner, and the PDB and Data Archiving curriculum. Our next IQB RCSB Protein Data Bank crash course will take place in the fall on October the 12th. The focus will be leveraging RCSB PDB application programming interfaces for bioinformatics analyses and machine learning. And we're targeting this virtual crash course to both bioinformatics and structural biology researchers, those who need to cross-reference PDB data and data from other trusted external data resources, and anyone interested in large-scale analyses of 3D structured data, both experimental and computational. More details will be available on both the rcsb.org web portal, our research-focused website, and on the Institute website, iqb.rutgers.edu. We also have opportunities for scientific software developers, both graduates and undergraduates. We're currently searching at Rutgers for a database administrator. We are always interested in getting applications for undergraduate summer researchers at Rutgers and gap year opportunities. These are particularly aimed at individuals with computer science training with an interest in biology in 3 dimensions. We are actively recruiting for a postdoctoral researcher at Rutgers with an expertise in metalloproteins, metalloenzymes, and always interested in the possibility of attracting additional postdoctoral researchers to our team at UCSD at the San Diego Supercomputer Center, where the focus would be bioinformatics. I want to begin by acknowledging our partners in the Worldwide Protein Data Bank, many of whom are depicted in these various photos, and you can see the logos of the full members of the Protein Data Bank, of the Worldwide Protein Data Bank, shown here on the bottom. Not included is our latest addition, the associate member from PDB China, which is based in Shanghai. And finally, the RCSB Protein Data Bank team includes a very large number of people who help ensure that we can deliver PDB data augmented by more than 1 million computed structure models to PDB data consumers worldwide, many millions of students, educators, researchers, and their trainees. I want to make specific and special mention of the late John Westbrook, who passed away tragically in 2021. He was one of the inventors of the PDBx and mmCIF data standard, and its champion literally until the day he died. We are funded for core operations by the National Science Foundation, 3 institutes at the National Institutes of Health and the United States Department of Energy. I'm grateful to our hosts at Rutgers, UCSD, and UCSF, and I'm grateful to all of you for your interest in the RCSB Protein Data Bank, your interest in the PDBx mmCIF data standard, and the PDB archive. Be safe and be successful. Thank you. So the webinar is now closing. I thank you again for participating, and look forward to seeing all of you and your friends and colleagues in October when we mount the second of these crash courses for 2023. Good luck, be safe, be successful, and stay away from COVID.